Firstly, good afternoon, everybody. I'm Kayleen, and I'm so excited to be sharing my research project with you all. It's entitled Functional Characterization of Expression Quantitative Trait Loci in ADME Genes. And just a little bit of background, pharmacogenetics is the study of varying responses due to pharmacological agents due to an individual's genotype. As you can see on the image on the right, 4 patients can receive the same medication but respond differently because of their genotype. Some can respond to a normal dose, some need a lower or higher dose, and others need a completely different medication. And this is all because of mutations or single nucleotide polymorphism, known as SNPs, in genes involved in that drug's absorption, metabolism, distribution, and excretion, which obviously impact the patient's drug sensitivity, resistance, or toxicity. In addition to these mutations, variations in other regions of the genome that regulate expression of the wild type or variant alleles of those ADME genes are a focus in pharmacogenetics. An example of this is, and the center of my project is eQTLs, which is a DNA locus that contains variants resulting in differential gene expression levels. The main problem faced in Africa right now is that a majority of the clinical trials and pharmacogenetics studies have been performed in cohorts of European, Asian, or African American ancestry. So the results show an array of variants which affect the efficacy and toxicity in treatment regimens, but the findings aren't useful in African populations since there's a knowledge bias towards pharmacogenetics-based interventions for non-African populations. This is problematic since novel alleles can be found in African populations and are not taken into account in clinical trials or drug safety guidelines. An example of this is HIV treatment, 5-FU, that's widely used in the continent, and its toxicity differs in patients due to varying levels of expression associated with the repeat number in the promoter of the TYMS gene. In Africa, 2 novel alleles were found which have more repeats, and those are found in other populations, and they could explain why there's a disproportionate amount of toxicity experienced by African patients who are exposed to 5-FU. So we hypothesized that genetic variants found in African populations may harbor eQTLs that result in differential gene expression. In addition, these eQTLs associated with ADME SNPs exert their regulatory control in an allele-specific manner. This may have functional consequences which significantly impact gene expression and therefore drug efficacy. This project therefore aims to functionally analyze eQTLs in association with genetic variants in a selection of ADME genes within African populations. The method I'll be using is cloning the wild type region of selected ADME genes harboring these eQTLs of interest into a luciferase reporter vector, and this will allow for the determination of reported gene expression levels. On the left, it's just a quick workflow of molecular cloning. So we have our luciferase reporter vector, which has the luciferase enzyme gene. It has ampicillin for selectable marking. And we can basically take our PCR insert of the ADME gene promoter, which harbors the eQTL of interest. We use restriction enzymes, clone it into that vector, perform a transformation, our colony screening, and those colonies that do have the insert will then move on to a luciferase assay. And as you may already know, the output of luciferase assay is luminescence, which is it directly speaks to protein expression. Moving on to the second objective, we'll perform site-directed mutagenesis to introduce the eQTL into the genomic region and assess its effect on reported gene expression in comparison to that of the wild type. This will show whether the eQTL has an effect on the expression level of the gene or not. The impact of the study is that the functional characterization of eQTLs in association with genetic variants in these pharmacogenes will enable the analysis of their impact on gene expression and drug efficacy. And these findings can be used to create guidelines for safe drug use or use of alternate drugs in the continent and its individual populations. I'd just like to acknowledge my supervisor for her support and guidance, and then my colleagues at the Functional Genetics Research Lab at the University of the Witwatersrand. And that's the end of my presentation. Thank you.
Thank you very much, Kayleen. Just a quick question. You mentioned you're using African populations. Do you know which populations you'll be using specifically?
So the eQTLs that were chosen, they were chosen by a PhD student who did the bioinformatics analysis, and she chose it based on the frequency of like very important pharmacogenes in the continent, well, obviously for the world, but then the ones that had significantly different frequencies within African populations. So I don't think that they're specific populations, but just those that are significantly different in the continent compared to that of other populations.
Okay, thank you. George has a question. He says, are you starting with a known list of ADME eQTLs, or are you exploring a selected set of African variants?
There is a list that was summarized by the student I just spoke about, and most of them are focused on the database from GKB, the ones that they decided were most important pharmacogenes.
All right. Are you by any chance working with Jenny Matthew?
Yes.
Okay. We know her very well. She's from our department.
Okay, great.